Hello everyone, can you hear me? Can you hear me?
Yes, ma'am.
Right. So I think we were discussing about proteins in the last class, right? So today we are going to discuss about the nucleic acid. Okay, so we are going to discuss about the nucleic acids. What are the nucleic acids which you know? Which are the nucleic acid which you people know?
DNA and RNA.
Right, DNA and RNA. So these are what? DNA, RNA, these are what? These are polymers of something, right? Polymers of nucleotide. Okay, these are polymers of nucleotide, right? So what do you mean by this nucleotide? What do you mean by this nucleotide? If you take a nucleotide, what is this nucleotide means? It is a compound having three groups, right? It's a compound having three groups. What are the three groups in a nucleotide? The three groups,
Nitrogen,
right,
pentose sugar,
right, pentose sugar and phosphate group. Okay, it has a nitrogenous base, pentose sugar and a phosphate group, right? If you take the nitrogen base, okay, or the nitrogenous base, what are the nitrogenous base? What are the two types of nitrogenous base? Right, it has purine and pyrimidine. Can you tell me what is the difference between this purine and pyrimidine? It differs in their structure. Okay, it differs in their structure. If you take pyrimidine, it has a pyrimidine ring. Okay, it has a pyrimidine ring. What do you know about this pyrimidine ring? It is a 2 carbon plus 4, sorry, not 2 carbon, it is 4 carbon plus 2 nitrogen ring. Okay, this is a 4 carbon plus 2 nitrogen ring, that is called as a pyrimidine ring, right? But if you take a purine molecule, it has two rings. Okay, one pyrimidine ring, that is the 2 carbon plus 3 nitrogen ring. Along with that, it has an imidazole ring. Okay, it has a imidazole ring. Understand? A pyrimidine ring along with an imidazole ring, that is called as a purine molecule. Okay, so they differ in their structure. Okay, in their structure. Okay, so the pyrimidine ring, I told you, it is 2 nitrogen plus 4 carbon. Then the imidazole ring, it is 2 nitrogen plus 3 carbon ring. Okay, right? So this is the structural way, the difference between purines and pyrimidines. Now give me the examples of purines and pyrimidines. This is important. What are the examples of pyrimidines?
thymine and cytosine.
Thiamine, cytosine. One more. Uracil. Thiamine, cytosine and uracil. What about purine? Adenine and guanine. Okay, adenine and guanine comes under purines, whereas pyrimidine, thymine, cytosine and uracil comes under pyrimidines, right? This is about the nitrogenous base, right? Now we are moving on to the second group, that is the pentose sugar. What do you know about the pentose sugars? Before that, I usually get doubt or usually get this type of question. What is the difference between pentose sugar and pentosaccharide? Pentose sugar and pentosaccharide. Anyone? What is a pentose sugar? Pentose sugar is nothing but a 5 carbon containing sugar. Okay, a sugar which has 5 carbon in that. But what about a pentosaccharide? A pentosaccharide is a compound which has 5 monosaccharide unit. Okay, it has 5 monosaccharide unit. Understand the difference? Pentose sugar is a single molecule of sugar which has 5 carbon in it. Okay, whereas pentose saccharide means it is 5 monosaccharide unit, right? The monosaccharide unit may have 3 carbons, 4 carbons, 5 carbons, 6 carbon. Okay, it depends upon the monosaccharide units, right? See, such 5 monosaccharide unit joins together to form a pentosaccharide. I think everyone is clear about the difference between pentose sugar and pentosaccharide. Okay, any doubt? Right? So tell me, what about the pentose sugar? What are the types of pentose sugar? Two types of pentose sugar, right? One is ribose sugar. Second is deoxyribose sugar. Okay. The ribose sugar is seen in where? It is seen in RNA. And deoxyribose sugar is seen in DNA. Right? Any doubt? Any doubt?
No doubt.
Right? So what now I'm going to do is I'm going to join this sugar nitrogenous base plus the pentose sugar. And what's the product which I get? What is the product which I get?
Nucleic acid.
Right, I'll get a nucleoside, S-I-D-E, nucleoside. Right? And what's the linkage between nitrogenous base and pentose sugar? What type of bond? These are the questions which you will get.
Hydrogen.
What type of bond between? It is the glycosidic bond. Okay, it is the glycosidic bond. Okay, there is a glycosidic bond between the nitrogen base and the pentose sugar. Understand? Understand, everyone? And can you give me an examples for this nucleoside? Examples for nucleoside. Adenosine, guanosine, thymidine, uridine, and cytidine. Okay, adenosine, guanosine, thymidine, uridine, and cytidine. These are the examples of nucleoside, right? So we were discussing about nucleoside, and the linkage between the nitrogenous base and the pentose sugar is glycosidic bond. Okay, or glycosidic linkage, right? Now I'm going to add one more group, nitrogenous base plus pentose sugar plus the phosphate group. What I get? What's the product? It is the nucleotide, right? Nucleotide. Understand? Nucleotide. Now, what's the linkage between the pentose sugar and phosphate group? What is the linkage between the pentose sugar and phosphate group? It is the ester bond. Okay, ester bond between the pentose sugar and phosphate group. Okay, and this ester bond is formed with the 5th carbon atom. Okay, it is formed with the 5th carbon atom of pentose sugar. Okay, the phosphate group will form ester bond with the 5th carbon atom of pentose sugar. Okay, very, very important. Understand? Ester bond is formed between the 5th carbon atom of pentose sugar, right? Now, what are the examples of nucleotide? Examples. It is adenylate acid. Okay, then guanylic acid, thymidilic acid, okay, uridylic acid, and cytidilic acid. Okay, this is the example of nucleotide. Okay, right? Now, first I am going to talk about the DNA. Tell me which are the nitrogenous base present in the DNA, or which is the base that is absent in DNA.
Uracil is absent in DNA.
Right, uracil is absent. So if you take the purines in DNA are adenine and guanine. Okay, then pyrimidines are, it is cytosine and thymine, right? Thymine and cytosine, right? And the sugar molecule is deoxyribose sugar. Okay, deoxyribose sugar, right? Now, who discovered this DNA, or who identified this DNA first? Who isolated this DNA? It was discovered or isolated first by Friedrich Mischer. Okay, he actually first isolated the DNA, right? But who observed the DNA, or who study about this DNA? Who first study about this DNA? Hmm? Who did a proper study? It was done by Wilkins. Okay, Wilkins and Franklin. Using which technique? Using X-ray crystallography. Okay, crystallography. Okay, so using the X-ray crystallography, Wilkins and Franklin study about the structure of DNA. Right now, we will see what they studied. Okay, when they studied, they came to know that DNA was, okay, before that, based on this study, somebody proposed a model for DNA, right? Based on this X-ray crystallographic study, not crystallograph, it is crystallography study by Wilkins and Franklin. Two other scientists, okay, proposed a model for DNA. What is the name? It is the double helix model, right? Double helix model. And that was proposed by whom? Right, by Watson and Crick. Okay, for that they have been awarded Nobel Prize. Okay, right? So they proposed this double helix model. Right now, if you take this double helix model, you know, we can see there are two complementary strands of DNA. Okay, what do you know about comps? No, that is right. These two complementary strands are anti-parallel. Okay, they are anti-parallel. But what is mean by this complementary strands? To know about complementary strand, you need to study about the pairing of nitrogenous bases. Okay, pairing of nitrogenous bases, right? Adenine will, you know, this adenine, the nitrogenous base are very stubborn. Okay, this adenine is very stubborn. It will goes only with thymine. Okay, it will goes only with thymine, right? Similarly, the guanine, they will go only with cytosine. Understand? And what is this line that I have drawn for adenine and guanine? Right, they are the hydrogen bonds, okay, which keep them together, right? So in between adenine and thymine, you have two hydrogen bonds, and in between the guanine and cytosine, you have three hydrogen bonds, right? This pairing of nitrogenous base, right? That in one DNA strand, if you have adenine, can you guess what will be the nitrogenous base in the next opposite strand? It will be thymine, right? Understand? Because I told you adenine will only goes along with thymine, right? Similarly, if you have guanine cytosine here, the opposite strand will have guanine, right? This is called as a complementary pair, right? If I can write the sequence of the nitrogenous base in one pair. Okay, I'm writing the sequence of nitrogenous base in one pair or one strand. Can I guess the nitrogenous base in the next strand? Can I guess what will be the nitrogenous base that is present in the opposite strand? Can I guess? Hmm? Yes, that is called as complementary pairing. Understand? If I have a sequence of strands in one, or sequence of nitrogenous base in one strand, the opposite strand or the anti-parallel strand, it will have the sequence of nitrogenous base complementary to it, right? Adenine along with thymine, cytosine with guanine, guanine with cytosine, cytosine with guanine, thymine with adenine, right? This is called as a complementary pairing. Okay, right? And what is this this opposite anti-parallel? Hmm? What is this anti-parallel nature? Hmm? This anti-parallel nature is said according to the direction of the phosphodiester bond. Okay, where you can see this phosphodiester bond? Where you can see this phosphodiester bond? Hmm?
Phosphate group.
Between the phosphate group and pentose sugar, right? And I told you this phosphate group will goes with fifth carbon of the pentose sugar. Understand? Understand?
Yes, ma'am.
Right. So if you draw the structure, okay, if you draw the structure of the DNA, if the direction of this phosphodiester bond, okay, it is from 5 dash to 3 dash direction. Okay, this dash represents the carbon atoms. Okay, what is this 5 dash 3 dash? I will explain. Okay, don't worry about that. I'll clearly show what is this 5 dash and 3 dash. Okay, or if you refer back to your video, you can understand what this 5 dash 3 dash means. Okay, so if the direction of phosphodiester bond in one strand is in 5 dash to 3 dash direction, then the next strand, the opposite strand, will have the phosphodiester bond in 3 dash to 5 dash direction. Understand? Right? That means if this strand start from the 5 dash end, it will ends at which end? Opposite end is the 3 dash end. Then this strand will start from 3 dash end and ends in 5 dash end. Okay. This 5 dash and 3 dash, it represents the carbon atom in the pentose sugar. Okay, in the pentose sugar, right? Just I'll just draw it here. If this is the pentose sugar, okay, and this is the fifth carbon which has the phosphate group. Okay, so this is the 5 dash carbon. Okay, right? If you take the next group, if you take the below one, the pentose sugar just below it, this is the fifth one, right? So actually this is like this. Right, so it will have a bond like this. Okay, this is the 5 dash 3 dash direction. Understand? Understand the phosphate linkage? This is the fifth carbon of the fifth carbon atom of the first sugar. If this is the second sugar, there is the bond between third carbon atom and the phosphate group. Okay, this is the phosphodiester bond. Okay, this is one ester bond, this is the next ester bond. Okay, phosphodiester bond, right? And this phosphodiester bond, okay, this phosphodiester bond, that is sugar phosphate sugar, this forms the backbone of the DNA. Okay, this forms the backbone of, okay, backbone of DNA. Understand what is a phosphodiester bond and what is how it is formed?
Yes, ma'am.
Okay, so this is about the structure. Okay, now we'll continue. So if you take a strand, okay, This is one nitrogenous base. This is the second pair. This is the third pair. Okay. Now what is the distance between each pairs? What is the distance? It is around
3.4 amps.
right 3.4 Armstrong, right? So I told you the DNA is a double helix structure, right? It's a alpha helix. Okay, alpha helix, right? So if you take one turn of the helix, this is one turn of the helix. Okay, this is like the one turn of the helix. Okay, so this one turn of the helix will have how many base pairs? One turn of the helix will have 10 base pairs. Okay, 10 base pairs, right? So what is the length, total length of one helix? It is 10 into 3.4 Armstrong, right? That is the 34 Armstrong. Okay, 34 Armstrong. Understand? Understand everyone? 34 Armstrong. Okay, and this 34 Armstrong, this is called as helix length. Okay, this is called as helix length. Understand? This is called as helix length. Okay, right? Now I told you this adenine will pairs with thymine, cytosine will pairs with guanine, right? So if you take the number of adenine will always be equal to thymine, number of cytosine will always be equal to guanine. Understand? That means what is this adenine and guanine? These two are purines, right? These two are purines, and cytosine and thymine, these two are pyrimidines, right? So if you take, we can understand the number of purines will be equal to number of pyrimidines. Okay, that means number of adenine plus guanine will be equal to thymine plus cytosine. Am I right? Am I right or not?
Yes, ma'am.
Right? So if you take a DNA and if you count the number of adenines, the number of adenines will be equal to the number of thymines, right? Similarly, the number of cytosine will be equal to number of guanines. That means the number of purines will be equal to number of pyrimidines. And what does this rule? What is the name of this rule?
Char-Gas.
Right, this is called as the Char-Gas rule. Okay, Char-Gas rule. This is called as Char-Gas rule. Okay, and this will be universally constant. If you take any species, any organism, this will be seen, right? The number of purines will be equal to number of pyrimidines, right? Now look carefully. What I'm going to do is I'm going to divide this A plus G by T plus C. What is this? A plus G by T plus C.
Equal to 1.
It is equal to 1. What is this? This ratio is called as the base ratio. Sorry, not this one. This will be always be equal to 1. What I'm going to do is A plus T by C plus G. Okay, this ratio A plus T by C plus G, this ratio is called as the base ratio. Okay, understand? This will be always be equal to 1. This we know, because we know the number of purines will be equal to number of pyrimidines. That is Char-Gas rule. But look at this ratio. Adenine plus thymine by guanine plus cytosine, and that ratio is called as base ratio, and this will be a constant. It will be a constant for a particular species. Okay, it will be a constant for a particular species, right? Like example, like if you take the base ratio of humans, okay, just an example, okay, base ratio of humans is 2. Okay, if you take any human, their base ratio will be always 2. Okay, the human can be from any part of the world, but their base ratio will be 2, right? If you take the another species like dog, let their base ratio be 3, right? If you take any species of in any dog, that species will have the base ratio 3. Understand the difference between base ratio and Char-Gas rule? Understand? Any confusion in the base ratio and Char-Gaff's rule?
No.
Okay, right? So this is about the base ratio and Char-Gaff's rule. Okay, now coming back to the structure, we know each helix has 10 base pair and the helix length is 34 Armstrong. What is the diameter? That diameter means what? Distance between two phosphate. Okay, distance between two phosphates, two phosphate group, right? Here one phosphate, here one phosphate. This is the diameter. What is the diameter? The diameter is 20 Armstrong. Okay, the diameter is 20 Armstrong. Understand? Okay,
Yes, ma'am.
right? Now coming to the types of DNA. Tell me, what are the types of DNA? I have given you a box in the in your video lectures where I discussed about type A, type B, type C, type D, right? Where does this, on what basis this types came, or based on which all criteria we have divided it into different types? Based on the,
Based on direction of rotation. Rhomboid.
that is L and D, right? Alpha helix or beta helix. That is not L and D, that is alpha helix or beta helix. The rotation is towards right side, then it is alpha helix, towards left side it is beta helix. That is right. But now I am asking you about the A, B, C, D types. This division is based on the helix length and number of base pairs in each helix. Okay, understand? This division is based on the helix length and the number of base pairs in each helix. Now can you tell me which type is human DNA? Human DNA is which type? A, B, C or D? Tell me. Human DNA comes under A, B, C or D category. Right, human DNA is B type. Right, it is B type. Human DNA is B type. Okay. Now, what is the function of DNA? Function of DNA. Storage of genetic information. And this is the only storage. This is the unit of inheritance, right? This is what is passed down to generations. Okay, unit of inheritance, right? The gene that is present in DNA is passed down from one generation to another generation. Okay, right? Now we'll move on to the next nucleic acid, which is the second nucleic acid which we want to discuss. That is RNA, right? Ribonucleic acid. Okay, here what is the difference? You have uracil instead of what? Right, instead of thymine you have uracil. Okay, instead of thymine you have uracil. And pentose sugar, which is the pentose sugar? Ribose sugar. Okay, ribose sugar. Okay, now what is the different, or what are the types of RNA? Types of RNA. Broadly, I can say I can divide the RNA into, right, genetic RNA or genomic RNA and non-genomic or non-genetic RNA. Where you can see this genetic RNA, or what is this genetic RNA? Right? In some organisms, instead of DNA, their genetic material is RNA. Understand? In humans, our genetic material is DNA, right? But in some organism, they have genetic material as RNAs, right? Can you give me an example for such organism?
virus.
Right? Reovirus. Okay, not all virus. Okay, not all virus. Some virus have DNA as their genetic material. Okay, so you should be particular, right? The reovirus. Okay, the tobacco mosaic virus. Okay, so these virus has RNA as their genetic material. Okay. Okay, not all viruses. Exclusively viruses doesn't have RNA as their genetic material. Some viruses have DNA as their genetic material. Okay, so when you say the answer as virus, be specific about the type of virus. Okay, that examples, two examples which is given in NCERT is reovirus and tobacco mosaic virus. Okay, right. Now coming to the non-genetic RNA. What are the types of non-genetic RNA? Yes. Bacteriophage, right? Some types of that is also that the bacteriophage, correct, RNA has the genetic material, but not all bacteriophage, right? This bacteriophage actually named in numbers. Okay, you can see 1 lambda plus 174 123. Okay, they are named in such numbers. Okay, bacteriophage. So some bacteriophages have RNA as their genetic material. Okay, like virus. Okay, not exclusively, not all bacteriophages have RNA as their genetic material, but some bacteriophages has RNA as their genetic material. Understand? Clear?
Yes,
Okay. Now coming to the, what are the non-genetic RNA? Types of non-genetic RNA.
not
Right. What are the types? rRNA. Correct. Then tRNA. Not transfer RNA it is. Okay. It is T means transfer RNA. Then mRNA or messenger RNA. Very good. The ribosomal RNA, what is the specialty? You know, the other two RNAs, the tRNA and mRNA, are synthesized inside the nucleus. Okay, both of these are synthesized inside the nucleus. Okay, both are synthesized inside the nucleus. Okay, synthesized inside the nucleus. But what about this rRNA? It is synthesized in the nucleolus. Okay, ribosomal RNA or rRNA are synthesized in the nucleolus. Okay, right? And what is their function? This is ribosome. Okay, small unit of ribosome and large unit of ribosome. Okay, and this ribosome, okay, this ribosomal RNA are seen inside these ribosomes. Okay, ribosomal RNA are seen inside these ribosomes, and this ribosomal RNA help in the attachment of the other two RNAs to the ribosomes. Okay, understand what is the function of ribosomal RNA? They are synthesized in the nucleolus. They are present abundantly inside the ribosomes. Okay, one more thing which I need to tell you is that 80 to 85% of all the RNA are rRNA. Okay, about 80% of all the RNAs are of rRNA type. Okay, and they are seen where? They are seen inside the ribosome, and they help in the attachment of other RNAs to the ribosome. That is the transfer RNA as well as the messenger RNA to the ribosome. Clear? Clear? Right? Now, what about the tRNA or transfer RNA? This will carry the amino acid, okay? Carry the amino acids during the time of protein synthesis, okay? They will carry the amino acid during the time of protein synthesis, okay? That is the function of tRNA, and I told it is synthesized inside the nucleus, right? Now coming to the mRNA. What is this mRNA? This is what you get from the DNA. Okay, if you have one DNA, right, if you take one DNA, what you get from the DNA during the time of protein synthesis. Okay, right, if you try to read DNA, right, and if you try to read DNA and produce an RNA, a corresponding RNA, okay, like TUAGC, right? If you try to produce or reproduce an RNA from a DNA strand, that process called as transcription. Okay, that the product which you get for protein synthesis is called as mRNA or messenger RNA. Understand? The RNA which is produced from the DNA for protein synthesis is the messenger RNA, and that process called as transcription, which you will study in detail in the next year. Okay, understand the types of RNA?
Yes, ma'am.
So can we just see some questions? Tell me. Nitrogenous base plus sugar. Nucleoside. Remember, plus sugar, S for S, nucleoside. Okay, nitrogenous base, sugar plus phosphate. It will comes as nucleotide. Okay, remember that T comes after S. Okay, so if there is 2, then it is nucleoside. If there is 3 components, then it is nucleotide. Okay, T comes after S. Okay, understand? Right, now the next question. Identify A and B. What is this A? This is A. This is B. This is 2 types of linkages are given, or 2 types of bond are given here. What type of bond is A? Between the pentose sugar and the nitrogenous base, that is adenine. Which is the bond? Glycosidic bond. Okay, glycosidic bond. Between A is glycosidic bond. And what is B? Between the 5th carbon. Here you have the 5th carbon. Right? It is the ester. So this is the 5th carbon. Okay, it is the ester bond. Okay, B is ester bond. Okay, right. Now next question. Please match. Thymidine. Thymidine is a nucleo. It is a nucleoside. It is a nucleoside. Thymidine. Guanylate acid. It is a nucleotide. Right, nucleotide. Cholesterol. Cholesterol is a lipid. Okay. Glycine. Glycine is it is the simple amino acid, right? It is the simple amino acid, right? So answer is A4, B3, C2 and D1, right? The backbone of DNA chain is formed by sugar, phosphate sugar chain. And what is this? Phosphodiester bond. Okay, phosphodiester bond, the sugar phosphate bond. Okay, sugar phosphate sugar, that's the phosphodiester bond, forms the backbone of DNA chain. Okay, understand? Nucleic acid. Any doubt in the chapter? So we have completed all the macromolecules. I think we have completed carbohydrate, we have completed lipids, we have completed proteins, and we have completed nucleic acid. Any doubt till here?
No doubt.
Okay, everyone. I'm hearing only one voice. That's why I'm asking. Any doubt, anyone? So in the next class, we will be discussing about the enzymes. Okay. No doubts? Then see you in the next class, everyone.